Hi, very good morning, one and all. Let us come to 10th class biology. Coming to 10th biology, present we are discussing about the topic reproduction, the generating system. So, according to the reproduction, already we completed almost 80% of the topic regarding the modes of reproduction like asexual and sexual mode of reproduction both in a plant and animal physiology. Is clear, children? Now, it's a very important concept regarding reproduction, that is cell division and continuation of life. It's a very important topic, cell division and continuation of life. So, what is meant by cell division? How the cell is divided into two equal halves. For example, one parent amoeba which gives rise to two daughter amoebae. Already we studied 8th class reproduction in animals, 4th chapter. So, one bacterium, that means one mother bacteria which gives rise to two daughter bacteria. So, that means one bacteria which gives rise to two daughter bacterium. Or one hydra which gives rise to two daughter hydra. So, all these binary fission or budding or fragmentation, as we learn about asexual mode of reproduction, is equal to reproduction in animals or plants or as prokaryotes. It is equal to cell division. So, our cell division is equal to reproduction in animals like a binary fission. Both are equal. So, when parent cell is divided into two equal halves means it is a binary fission which is more or less it is equivalent to that cell division. Our cell division is equivalent to that binary fission or asexual mode of reproduction as we are treating. So, let us come to the point. First of all, actually the cell division concept, we are having some basic information regarding about the cell division concept. The first one is cell division and continuation of life. So, when the gametes are fusion, it results to the zygote formation. It is in the diploid condition. For example, see, male gamete means sperm cell which are having a haploid number of nucleus. The female gamete like ovum which are having the haploid number of chromosomes. Already we studied in the sexual reproduction in animals as well as the plants. For example, pollen grains and ovules in the female plant physiology. While coming to the human physiology, male gametes like sperm cell and female gametes like ovum. Is it clear, children? So, by the process of fertilization or pollination, it results to the formation of zygote which are diploid to a number of conditions. This is the first and foremost step in the fertilization process. From there onwards, the cell divides continuously up to certain age, and we are having to develop entire the whole body like tissues, organs and organ systems. Finally, we have to make an adult. This is the mechanism which is behind the cell division. But how the cell can be divided? How many phases are there in that hiddenly in that cell division process? How the cell division makes a continuation of our life? It's very clear. So, once zygote formation, we start our life. But when we are maturing, for example, first mature we call as a menarche, means ovulation process. The females obviously they have to release one ovum. It's very important, children. So, at the time of ovulation, the girl child can release only one ovum which are haploid in number. That means the chromosome number is 22 pairs of autosomes. So that means 22 autosomes. Autosomes are 22 plus X. So this is the state of womb. This is the state of womb. While coming to the sperm, 22 autosomes plus X are by sperm cell. For example, I am taking the Y sperm cell. So, the combination is 44 plus XY. It is the combination of male child. Is it clear, children? So, entire the life, we are having the cell number like 46. But exclusively at the time of releasing of the gametes or the germ cells or the reproductive cells or the sex cells, the chromosomal number is haploid number by the process of meiosis. So, meiosis occurs in the sexual cells or germ cells or the gametes which results to the reduction division. Why it happens or else what will happen? So, the reduction division means from the female parent 44, out of 44 autosomes only 22 autosomes in the ovum. Out of 2 ovumes only one ovum, that means one X ovum comes from the female parent side, as well as the male parent out of 44 autosomes, only 22 autosomes. Out of X or Y sperm cell, only one sperm cell will come and fuse with the other gamete. Why? Because our chromosome number is always remain constant in every successive cell division. Is clear, children? Or else the chromosome number is double in every generation. It becomes to abnormal shape and size. Our species also will lost by these mutations. All of sudden changes. Abnormalities in the autosomal or sexual chromosomes. Is clear, children? So that meiosis takes place to maintain the constancy of a chromosome number, and mitosis means every successive cell division the chromosome number is always constant. Why? Because see, it occurs in the somatic cells, like all the cells except gamete cells. All our bodily cells are called as somatic cells or physical cells. So it is having the equal number of chromosomes. It maintains the equal number of chromosomes. For example, parent amoeba is having one nucleus. The daughter amoeba, two daughter amoebas are also having the same number of chromosomes. Means single number of chromosomes, single number of chromosomes. How it is occurs? That is called as mitosis, doubling of chromosomes. And they are sharing the chromosomes into two equal halves. This is called as mitosis. So before coming to the mitosis or meiosis, like the cell division process, first we have to go to the history of or the process of cell division, the contribution of the different scientists, which plays a vital role to the conclusion part of the continuation of is life by the cell division process. Is clear, children? So among the scientists, first and foremost one is Schleiden. So it is a very familiar scientist. Schleiden is a very familiar scientist which is related to plant cell discovery. So in 9th class first chapter, cell structure and functions, regarding the nucleus, he is given the statement about so he discovered about that plant cell, as well as he treated or he is considered that nucleus is called as a cytoblast. Cytoblast means the other name of nucleus. Of course, we are called as nucleus is the boss of the cell or the control room, or it is also called as head of the cell. It carries the genetic material or genetic information. So nucleus is called as a cytoblast. Next. So I mean that Schleiden is having the knowledge about the nucleus involved in the cell division. So that he is called as cytoblast. Next, cell theory. According to the Schleiden and Schwann discovered the animal cell, Schleiden discovered the plant cell. So Schwann discovered the animal cell. So according to the cell theory, the second important cardinal principle is all the new cells are arising from the already pre-existing cells. So how the new cells, how the new gametes will come, how our offspring will produce. So already existing parents. So the parents which gives rise to gametes. So without parents, there is no offspring. Why? Because all the new cells, all the new gametes are formed from the already pre-existing cells. So this statement of cell theory was given by Rudolf Carl-Virchow. Rudolf Carl-Virchow has given the statement which is regarding to cell theory. All the new cells are arising from the already pre-existing cells. So he given the phrase means a combination of a word which is called as a statement. So omnicella, decella, he is given the phrase of this omnicella and decella. Next, important concept regarding this cell division and continuation of life. First and foremost person is German scientist like Robert Remarque in 1852. Robert Remarque observed that binary fission. For example, binary fission in amoeba or euglena or paramecium or whatever it may be like bacteria, it is equivalent to the reproduction of animal cell. Reproduction of animal cell is also called as a binary fission. Next, another important scientist in 1879, Walter Fleming. So he plays a crucial role regarding this cell division process. So he observed that the string-like structures in the nucleus. So we know about the structure of nucleus. This is called as a nuclear membrane and nucleolus. We are having the string-like structures are called as chromosomes or chromatin. The network-like structures are called as chromatin or chromosome. The number of chromosomes are constant. Those are called as 46 in number or 23 pairs in human beings. Is clear, children? So he observed that the string-like structures in nucleus split longitudinally during the cell division. So the these string-like structures, just we have to take this. This is a chromosome, X chromosome. So this chromosome is having the string-like structures. This we call as arms or chromatids. So they have to split longitudinally. They have to split longitudinally. So these chromosomes are split. So he observed that the chromosomes are split longitudinally and during the cell division. Next, he named such process of division. Is called as mitosis. Mitosis means a fine threads. It is a fine thread-like structures. These are called as chromosomes. He also called as these structures are called as chromosomes, means colored bodies. So chromosomes are nothing but colored bodies. So when we have to observe the diagram, see here, they are given to the colored string-like structures. These fine thread-like structures are called as colored bodies. These colored bodies are also called as chromosomes. Chromosomes carries the genetical information through the DNA or RNA, which are having the genes. This already I given the basic information about genetics. See children, so whenever the chromosomes are split longitudinally and they are going to share these chromosomes into the daughter cells. So that is why cell division takes in 4 phases. And he also observed that it can occurs in a sequence of a process that is called as prophase, metaphase, anaphase and telophase. So once again I repeat the contribution of Walter Fleming regarding cell division and continuation of life. First he observed that the string-like structures which are present in the nucleus is called as chromosomes, or fine threads he called as chromosomes. So he named such process of division is called as mitosis, means fine threads, and moreover he also observed that it has occurs in a sequential order. Sequential order means the mitosis occurs in 4 phases: prophase, metaphase, anaphase and finally telophase. After completion of these 4 phases, one parent cell which split longitudinally and it can share to the two daughter cells. So one chromosome is divided into two chromosomes. So this is occurs in 4 phases. 4 phases. This is called as a mitosis. So as a result of mitosis, the chromosome number is double, but the number of chromosomes are doubling, but the same number of chromosomes. So the daughter cells are 2. But the chromosome number is the same. The chromosome number is same. Only one chromosome in every daughter cell. So I mean that every amoeba, or every daughter amoeba, or every daughter bacteria is having same number of chromosomes. So that each is equal to the parent cell. Clear children? So a decade later, after a decade means in 1880, almost 1890s, a decade later, these thread-like structures are called as chromosomes. We call fine threads like mitosis. So afterwards it is called as a chromosome, means it is a colored bodies. So the fine threads are having the colored bodies. Those are called as chromosomes, which is clear children. Next we are coming to the another person, Wilhelm Rocks. So Wilhelm Rocks is proposed that the chromosomes could carry out the heritable elements. So these chromosomes are having some genetical information. So they carries some genetical information, the hereditary elements like this DNA, RNA, like nucleic acids and genes. So those days we called the word heritable elements. Later it is called as genetics or genes. So genes which are related to genetics. So regarding genetics, Gregor John Mendel, who has carried out a number of thousands of experiments under the Gordon P. We have to explain about detail in this Mendel genetics in a 8th chapter, heredity. One more chapter after we are going to discuss about in a detailed manner about Mendel's experiments on the Gordon P. తోట బఠానీ మొక్కని చేసినటువంటి ప్రయోగాల గురించి మనం 8వ చాప్టర్ అయినటువంటి heredity means inheritance. Heredity and inheritance we are going to explain. Next, in cell division, the cell divides into 2 equal halves with equal number of chromosomes, which are similar to the parent cell and diploid in the nature. It's very important. So during the cell division, what will happen? Once again I repeat. So in cell division, the cell divides into 2 equal halves as we shown. For example, see in the cell division, one cell is divided into 2 equal halves. Next, equal number of chromosomes. When they are splitting this, one cell is divided into 2 daughter cells which are having the same number of chromosomes. See here 4 number of chromosomes, 4 number of. The chromosome number is the chromosomes which are similar to the parent cell and are deployed in the nature. See, each and every cell, for example, see in this schematic diagram of mitosis, whenever we have to observe, so every cell is in a diploid state, that means 2n condition. Every cell is a diploid condition except the germ cells. Is clear, children? So it is occurs by this mitosis process. Actually, but the chromosomes number is always constant. For example, one cell which can be divided, the chromosome number is decreasing, but not here. The chromosome number is always constant. This is quite an amazing concept regarding the biologists. So biologists are also wonder about this concept and they have to begin to study. So apart from that, one of the person is called as August Weismann. So August Weismann, a biologist, he hypothesized that if suppose in every successive cell division, for example, they are deployed in the nature. He said that deployed in the nature. That means every cell is deployed in nature. Means, for example, these germ cells, sex cells are also deployed. 2x, that means, for example, ovomes, 2 ovomes are released. In sperm cells, X sperm cell, Y sperm cell will come. The autosomes like 44 plus 44. So in every generation, the chromosome number is double, but not like that. The chromosome number is always constant. Means we are having only 46 number of chromosomes in every successive generation, cell division. So he hypothesized that meiosis occurs in the sexually cells. So that our chromosome number, as said, so half of the chromosomes are only comes to the gametes. Only half of the chromosomes are comes to gamete cells. Is clear, children? So this is happens. He explained about. So he hypothesized that. So according to his statements, in successive generations, individuals of the same species have the same number of chromosomes in a successive cell divisions. Why we use that successive cell divisions? For example, in some abnormal, we are having some chromosomal abnormalities like autosomal abnormalities and allosomal abnormalities. Is it clear, children? That I have to explain in the next. Next, in successive cell division, the number of chromosomes always remain constant. In every successive cell division, the number of chromosomes is always remain constant, and the same number of chromosomes in that same species. For example, human beings always 46. So every successive cell division, 46 chromosomes only will be present in that every daughter cell. It is clear, children? So this is the information which was given by this the scientists which are related to Schleiden or Rudolf von Virchow, next Robert Raymond, Walter Fleming, next William Rox, next August Weismann. So these are the scientists which are given the related information regarding about cell division and continuation of life. Then we are moving to the schematic representation was given by the Theodor Boveri. So the schematic diagram which are related to mitosis. Mitosis means fine threads. So these colored bodies like the fine threads, So these are called as fine threads, which involves in the 4 phases means 4 stages. Prophase, first stage. So the decondensed state of these chromatin are comes to that together, and they have to the colored bits like threads. These are called as chromatids. Next, they are having a centriosome. See, center they are having the centriosomes, and they are having the spindle fibers. Spindle fibers means kandetantu. So north pole and south pole they are having the spindle fibers. So with the help of spindle fibers, see, all the chromosomes are having the middle plate. The centriosomes are comes to middle plate. So with the help of these spindle fibers, of north pole and south pole, they have to pull out. So for example, see, imagine that this is a chromosome. So these chromosomes are middle. So when with the help of spindle fibers, they have to pulling to the south pole and north pole. So they have to split. See, this is a chromosome, which is a splitting over. Next, they have to form the remaining part. So this remaining part is formed by this sharing of this chromosomes. So this is called as telophase. So prophase means the decondensed state of chromatin comes to condensation and disappearing of the nuclear membrane, nucleolus. The chromosomes are comes to closer and they have to become a fine thread like structures. That is the prophase. So metaphase means the centriosomes are comes to middle plate or equatorial plate and they are having the spindle fibers or appears. So with the help of these spindle fibers, north and south poles they have to split in a longitudinal section. So this is called as a longitudinal section. So with the result of splitting of this longitudinal section, they have to become to double. So these double number of chromosomes are splitting, sharing to the two each daughter cell. So this is occurs in the telophase, Chalanadasa, moving stage. So in this moving stage, they have to move to respectively one and the other. When the karyokinesis means nucleus is divided into two daughter nuclei, then following by that cytoplasm, that is called as cytokinesis. So finally, with the help of this schematic representation given by this Theodore Boveri, the schematic representation about this mitosis. So prophase, metaphase, anaphase, telophase. Just you have to go through the textbook page number 136. You have to read out this total table and we have to understand about that the clear information regarding about prophase, metaphase and anaphase and telophase. Next class and next video we are going to explain about meiosis. What do you mean by meiosis and why it is called as a reduction division, why it is called as a equal division, equal number of chromosomes, and how the gametes are having the reduction division by this meiosis. Thank you children, thank you for watching this video.